Hello everyone. Today we are going to see about nanodrop. Since this nanodrop is a very essential instrument that we use in all the laboratories to check the purity of our samples. This is a spectrophotometer that quantifies your samples containing DNA, RNA and protein with a very minimum quantity. For example, 1 or 2 microliters is more than enough to measure the concentration of your sample, and it gives us the concentration and purity of your sample. It gives the concentration in nanogram per microliters, and it helps us to measure the purity according to the ratio which is observed by the ultraviolet observance of three different wavelengths: 230, 260 and 280 nanometers. This 260 to 280 nanometer ratio gives us how pure the sample is from contaminating from the proteins. Since the protein is observed at 280 nanometers, the optimal ratio for DNA is 1.8 and for your RNA is 2. If your sample is less than 1.7, just simply avoid using the sample because it will inhibit many downstream applications. This 260 to 280 ratio shows us how pure our sample is from salts and other contaminants, for example, EDTA, phenol, etc., which are observed at 230 nanometers. The optimal ratio for this for both DNA and RNA is slightly 2 or above 2. Anything less than 2 suggests that there are some other factors in the sample which may also affect your further results. In order to improve the ratio of 260 to 280, which are contaminated by your proteins, you can treat your sample with more proteinase K enzyme and incubate it overnight. To improve your 260 to 230 ratio, you can clean your samples using spin columns, or you can increase the washing steps, or you can reprecipitate them to remove the contaminants. However, by using these improvement steps, you can also lose some of the nucleic acid concentrations. So, if your concentration is very low, you can start your experiment from extracting the whole sample from the source. Along with the purity ratio, this instrument will also give us a curve. This curve helps us in determining the presence of your nucleic acid. Since your DNA and RNA are observed at 260 nanometers, you should see a presence of only one nice peak at 260 nanometers and no peaks elsewhere. If there are double peaks or there are shift in the curve away from your 260 nanometers, then that your samples are not pure. I'm here to suggest you some of the tips to use for the nanodrop. Always clean the pedestal before use. Use distilled water onto the lower pedestal and close the upper pedestal. Leave these for 1 minute before wiping both the pedestals with lint-free tissue. Always measure your blank. You need to ensure your blank is exactly that, a sample free from nucleic acids and contaminants. If not, your sample value will suffer. Check for air bubbles. Take care when pipetting and ensure all the sample is deposited onto the center, the black dot at the bottom pedestal. Also, place the pedestal down on the sample slowly, taking a little extra time to ensure your sample is bubble free is a good practice. You can also be very sure by mixing your samples before measuring. You should apply mixing to all of your samples regardless of their prior state. For example, if you are taking a sample out of a freezer, make sure that the sample is completely drawn and is homogenized by a brief vortexing it before you use it for measurements. Be wary of your low concentration samples. Reblank your measure for at least every 30 minutes and take measurements in duplicate at least to make sure that the sample is homogeneous by mixing. I suggest you always write down your results. By writing down them, this keeps a paper copy of your result instead or in case if your machine breaks or something may happen to your results. Thank you.